Thanks, Chris. Great talk, and congratulations on very innovative concept for use of nanotechnology and delivery with the ability to simultaneously address targeting and therapy and reporting and dose there. Very, very creative. Our next speaker, and in fact our next two speakers, are from a institution which is actually just up the road from the headquarters of the conference sponsor, United Therapeutics, University of Maryland. And it was my great pleasure to meet Dr. Sengbak Lee about 1 year ago today when we held a joint United Therapeutics, University of Maryland Department of Chemistry workshop, and Sengbak Lee presented some really fascinating research, and I'm so glad that he's continued to develop that research and is here to report on it today. Dr. Lee has advanced degrees, including a master's degree and a PhD degree from Seoul National University. He's currently an assistant professor on already being promoted to tenured associate professor at the Department of Chemistry and Biochemistry at the University of Maryland. And that no doubt reflects both his great teaching abilities and his great research ability. So very congratulations on that. Dr. Lee's research focuses on the synthesis and control of nanotube structures, especially of some quite creative and unique selection of materials, which also shows his great ability to think a little bit outside of the standard box that everyone is using. And the title of today's talk reflects Dr. Lee's very trailblazing research. It's entitled Silica Nanotubes, Multifunctional Designer Nanoparticles at the Nano-Bio Interface. Please join me in bringing a warm welcome to Dr. Lee.
Thank you very much. It's a very kind introduction, and thanks for inviting me to give this nice opportunity to talk about my research here. Okay. Somehow it doesn't show. It's kind of cut it out here. And when you look at this subtitle here, nanotubes beyond carbon nanotubes, when I usually give talk out there, I usually use only the nanotube work because I'm basically a material chemist, and I'm making nanotube structures with various materials from metal oxide, metals, and conductive polymers and other polymers and various things. Oh, thank you very much. So it is very common that people think about this as a carbon nanotube talk when they look at the nanotube. So I usually use this subtitle, that nanotubes beyond carbon nanotubes. So today, I hope that you can get some information that there is another nanotube world beyond carbon nanotube. That is, I guess, the only thing that you can learn from my talk. Okay. Why nanotube? I will go over very briefly why nanotubes and what kind of method that we can use to make these nanotube structures, and also some of the nanotube applications, and which is including the magnetic nanotubes and also nanotube sensors. The later part I will not cover for that. Before I forget, there are so many students and postdoctors and also our collaborators. Without them, and also, of course, funding agencies. Without them, I cannot do all of these works. The reason why I'm interested in the nanotube structure is very simple, and everybody already talked about this multifunctionality. And when you look at the specific structures of nanotube structure, and I hope I would like to emphasize that you focus on this hollow nanoparticle structures in this case. And when you look at this nanotube structure, it has distinctive inner and outer surfaces, and also has an open end. And now, through this open end, you can control those many different drug molecules and also many other functionalities. You can put it in, also control the release. And our surface can be modified with targeting agent and also together with some environmental friendly molecules together. And one more thing that I should point out here is there are so many nanoparticles out there, but the controllability of those nanoparticle synthesis is one of the very important issue. So certainly in medical center, they would like to have just almost monodispersed particle. If they want 50 nanometer, then they want 50 nanometer. They do not want 70 or 100 nanometer together in that solutions. So that's why I came up with how do we control the shape of nanotube precisely, and that's why I chose this so-called template synthesis. Template synthesis technique is very, very simple. It has been already developed for more than 20 years, and the father of template synthesis is Charles Martin in University of Florida, and Charles Martin, Moskovitz, and Maluk groups are kind of the early pioneers in this template synthesis. And the concept is very, very simple. Imagine that you have a very well-defined porous structures. Actually, Dr. Halamki, previous session, talked about very briefly about the porous alumina membranes, porous alumina film structures. And that is also one of the example for those porous membrane or porous structures. But you have to note that these porous structures are very well defined. And also these porous structures are cylindrical here. So you take this porous membrane and dip into an interesting solution, take it out, dry, then you will see all those materials is coated on this membrane, including inner pore surfaces here. And then we move out the top and bottom layer. Then if you can selectively dissolve out the template, then you can have this nanotube structure here. And also certainly that there is an alternative route to grow this nanotube structure using some other electrochemical technique or other deposition technique. And now this is one of the example of porous alumina membrane. When you look at this SEM images, it's almost monodispersed size of pores. And when you look at the cross-section here, then each pore has very nice cylindrical structures. Certainly, the pore structure can be controllable. It's generally from 15 to 300 nanometer, but if you want to go down to 5 nanometer, then you certainly can go down to 5 nanometers using different ionization conditions. Also, the length of pore can be controlled generally from 100 nanometer to a few hundred micrometers. So the aspect ratio can be extremely short to the extremely high. Then what we are working on right now is these are those silica, one of the TM images and SEM images of silica nanotubes that we make. And using these silica nanotube based on those template synthesis technique, then what we can do is the chemical separations and also targeted drug delivery and bioimagings and also very fundamental studies related to the diffusion and wetting, also the reaction, various reactions in the nanotube structures, and also doing some sensing work with shape differentiated nanotube structures. So I will specifically will cover some of these separations and drug delivery and sensor work here today. The first important step to have multifunctionality in this nanotube, hollow nanotube structure is differential functionalization between inner surfaces and outer surfaces of nanotube. So this is one of the example what we did, and we put a lot of lots of dye molecules inside of nanotube and modify outer surface with specific proteins, and then let them interact with another specific biosubstrate, and then rinse them out. Then you see these individual nanotube is fluorescing on the specific biosubstrate. So this is another beauty of template synthesis. So while these nanotubes are embedded in that template, you can modify inner surfaces of nanotube, and then pulling out the nanotube and outside, and then modify outer surfaces with the second materials. So that is another advantage of using this template synthesis. So these days, we are really focusing on this magnetic nanotubes, and this, we made this magnetic nanotube by simply putting the magnetic particles in this silica nanotube. So this is a simple process of the synthesis of magnetic nanotube. You have a porous template and silica coating, and then release the, before you release the nanotube, you put these magnetic single crystals. You actually, not putting them, you actually synthesize directly in the nanotube these magnetic single crystals in this nanotube, and then release them out. And now you have a magnetic nanotube, magnetic property. You can have magnetic property on this nanotube. And when you look at this squid data, you see extremely high saturation magnetization value here, which is equivalent to those bulk magnetic particles. In this case, the magnetic particle is magnet-tight. So there are not many groups who can make this bulk magnetic saturation values with these magnetic particles. So one of the very interesting application, I guess somehow the picture has been, the PowerPoint has been a little bit destroyed by converting process. Anyway, one of the very interesting application of using this magnetic nanotube is toxic hydrophobic chemical separations. And out there in water system, there are so many toxic, extremely hydrophobic chemicals like PCBs and PAHs. And we want to analyze those toxic chemicals. But the problem is, since they are extremely hydrophobic, their concentrations are also extremely low. At most, it's a nanomolar concentration, very often picomolar or even femtomolar concentration. So current technology is using pretty large column, and you pour a few liters of water, capture them, and pour organic solvent, release them, evaporate, concentrate. Now you can take it out and put it in the instrumentation instrument to analyze them. So, however, using this magnetic nanotube, what you can do is we modify inner surface with extremely hydrophobic chemicals like a C18, and then modify outer surface with hydrophilic environmental friendly molecules, so that these nanotubes can, even though this nanotube has hydrophobic functionalities, but this nanotube can be completely suspended in aqueous systems and diffuse around to capture those hydrophobic chemicals. So for the proof of concept, instead of using real hydrophobic toxic chemicals, we use these extremely hydrophobic dye molecules to see the color. And you see this is the aqueous, this dihydrophobic dye solution here. You see red color. And then after putting this magnetic nanotube in here, applying magnetic field, you see red color move toward the magnet, which means this red color is not anymore from the solution, but from the nanotube. So after simply separating this red color with using this magnet, you see individual nanotube contains a lot of dye molecules inside. So right now, Agilent Technology is developing a pre-sampler using this technology to get those hydrophobic toxic chemicals. More important thing here is it's not just selecting those hydrophobicity, but also selecting the size of molecules because we can control precisely the inner diameter of the nanotube. Based on this one, you actually can select those small hydrophobic toxic chemicals rather than large. So you actually have a size of selectivity here. And in the same issue, you also do the bioseparations. Especially this bioseparation is pretty useful to separate those proteins that has dimers and trimers. So you actually can selectively extract out the monomers from the dimer and trimer mixtures. Again, we are also working on this drug delivery with using magnetic nanotube. And when you think of an ideal drug delivery carrier, and already a lot of in previous session, excellent speakers talked about this one, and they need drug molecules, imaging capability, targeting, and also material should not be toxic. Then this magnetic nanotube can contain those drug molecules inside, which is pretty important. One of the good reasons is many drug molecules, especially for cancer drug molecules, are hydrophobic. So imagine that you have those hydrophobic drug molecules on the outer surface of nanoparticles, and also if they cover more than 20% surface of these nanoparticles, outer surface, then this nanoparticle is not anymore really hydrophilic. And they will change its surface property dramatically. And this nanoparticle is likely to stick on pretty much everywhere through the nonspecific hydrophobic interactions. So there is a good reason why you need to hide and separate those hydrophobic drug molecules inside the particles and keep them outside surface hydrophilic and also environmental friendly. And another example for those reasons is if you want to deliver genes like DNAs and RNAs on outer surfaces, then those are exposed to the enzymatic actions. So those enzymatic actions may destroy and chop up those genes. Now then those genes is not anymore the same thing that you want to deliver. So that's why you need to separate and hide and protect those drug molecules in the nanoparticles. And again, also there is a possibility that you can control the movement of these magnetic particles using magnetic field-assisted immunobindings. Also, magnetic property can provide imaging capabilities. However, when you look at this TEM images of magnetic nanotube, these are magnetic nanotube here. This is about 50 nanometer diameter and 200 nanometer length, and relatively large compared to the other nanoparticles. So the first question that we had was, can this nanotube really be internalized into the cells, or they just stay on outer surface of the cells? And that was our first question. So we simply let them interact with the cells. Actually, these are breast cancer cells. And you see these green fluorescence, which is actually the nanotube itself. And since we put a lot of green fluorescent dye molecules in the nanotube. So, and this is confocal microscope image from the center of the cell, so which tells us that the nanotube can certainly be internalized easily to the cells. And this is after 30-minute incubations. And also, we did a little bit of a simple MTT assay for 3 days, and with cancer cells and some other red glioma cells, and they're non-toxic so far. This is the TEM image that shows that in the cells, you see these individual nanotubes here. Some of them are pretty much the good news is that some of them are founded in cytosol, which is good news for the drug delivery. And there is one laying down nanotube, which is extremely actually lucky in this case. When you look at this, the thickness of cell slice, which is 60 nanometer, then most of nanotube, then you cut this 60 nanometer, then most of nanotube should show this kind of a circle. But certainly, we were lucky to see this laying down nanotube here. Another thing is we wanted to know what is the mechanism for those endocytosis. Is it really endocytosis or the simple diffusion of nanoparticle into the cells? So what we did is the inhibition of endocytosis process by using chemicals. So there is a sodium azide or a sucrose to inhibit this endocytosis process. You see, in this case, this is breast cancer cell and this is HUVEC cell. And both cases, you see the great decrease of internalization of nanotube into the cells, which tells us that the main mechanism is endocytosis process. However, you still see some of the green dots in here, which is about 20%. That means these nanotubes are also using different mechanism to be internalized into the cells, which could be a simple diffusion. And for the MRI imaging, we label the cells with magnetic nanotubes and then measure the MRI imaging by increasing the magnetic nanotube concentrations in here. And this image, these are actually the T2-based image. And these magnetic nanotube is also good material for T1-based imaging too. But certainly the T2 has a lot higher value than T1, so we are showing this T1-based imaging in here. And this contrast is at around the level of less than 10 picomolar concentration of magnetic nanotube. When you retro-calculate back per cell, then usually 5 nanotubes per cell is the average number in this case. So which means in order to look at where these nanotubes are in the cells or around the tumor, that what you need is around 5 to 10 number of nanotubes per cell. So you don't need really lots of nanotubes per cell. And for the targeting images, this is also another work of Professor Martin's group, and not yet published, but we're going to publish. It will be came up soon. And for the targeting, which is targeting MDA-MB231 breast cancer cell, and what we did is we modified outer surface of the nanotube with a specific antibody that can target the cell surface receptor. And another control experiment is we also did a normal cell too, but this is another control experiment that we modified the outer surface of the nanotube with a different type of antibody that targets different cell receptors. But certainly this cell receptor, IGF-1R alpha, is overexpressed on this breast cancer cell. So certainly you see lots of nanotubes are on this in this case, but not a little bit of nanotubes are binding on these cell surfaces. So certainly it has a high selectivity and specificity in this case. But when you think of drug delivery, then certainly another big question is how you're going to control drug release and uptake and release rate. And certainly one of the easiest, probably the easiest example is simply you use the chemical interactions between the small drug chemicals and also inner surfaces of nanotube. Then you certainly can control some of the drug uptake and release rate. However, this may not be the ideal situation. Probably the ideal situation would be somehow you put a lot of drug molecules in these pores of the nanotube, and then somehow you clog them up in only the entrance of the nanotube, and then deliver them. Then somehow you open them so that those drug molecules can be released to in the target. So what we were trying to do was we were trying to control the diffusion of gold nanoparticles into the nanotube, so we could perfectly control the gold nanoparticle diffusions only at the entrance of the nanotube, and then use those gold nanoparticles as a seed to grow more gold to clog the pores of the nanotube. So this TM image is showing that gold nanoparticle is completely blocking the nanotubes, entrance of the nanotubes. And also using the same technique, we also could modify inner surface of only half of the inner surface of the nanotube with different chemicals. So which is another technique to introduce one or two or multiple chemicals into this nanoparticle surface, inner surfaces. And not only with using those conventional, just the straight porous structures as a template, we also can create these shape-differentiated porous structures in here in the template. When you look at that, the upper part has a bigger diameter and the lower part has smaller diameters. And we also can control the ratio in between. So using these shape-differentiated porous structures as a template, certainly you can create these shape-differentiated silicon nanotubes. So what does that mean? And very interestingly, previously, what you saw was just the shape-differentiated nanotube structures in TEM images or SEM images. Certainly, you can distinguish the shape. But more interesting thing is you actually can distinguish these a whole lot of different shapes of nanotubes through the conventional optical microscope, because you have huge differences in reflectance values between thicker part and thinner part. So these are conventional optical microscope images, and you see there's a SNT1, SNT2, a whole different type of nanotubes there, and each different shape of nanotube can act as a bio-labels or bio-code. So these are basically the dispersible bio-code, and using this whole purpose of using this shape-differentiated nanotube as a bio-code is multiplex sensings. So this is a very simple proof of concept experiment that imagine that you have an A-shaped nanotube and B-shaped nanotube here. You modify A-shaped nanotube with A proteins and B-shaped nanotube with B proteins. And you mix them together. Now you have a library with the different shapes of nanotube. And then you take a small aliquot of the solution and put it on the glass slide. You see there's an A shape, here's a B shape. And you know there is A protein on it, you know there is a B protein on it. Now you add simply a small amount of targeting molecules, analyte. Then if that analyte is B prime proteins that can interact with only B proteins, then only you see only B-shaped nanotube lights up. So just as a proof of concept, these are 3 different shapes of nanotube, and we add 2 different analyte here. You see only 2 nanotubes are lights up. So imagine that you have 10 different shapes of nanotube, then you can analyze 10 different analytes simultaneously at a time. That is the whole purpose of using this shape-differentiated dispersible nanosensors. And this is cross-talking, checking the cross-talking interactions, but you don't see any cross-talking interactions. And you certainly want to know how sensitive it is. Currently, we're using about million number of nanotubes and to get this about 10 picomolar detection limit. But certainly by decreasing the number of nanotubes per solutions, then theoretically we can get down to less than femtomolar concentrations. That is our detection limit. And you also want to know how many number of nanotubes can we create as a number of code. So certainly what you see here is that these are 4 segmented porous, differentiated pore structures. And this 4 segmented, using this 4 segmented differentiated pore structures, you certainly can create these 4 segmented nanotube structures. And these nanotubes can be created theoretically up to about 5,000 number of code. And certainly you can control the length of nanotube and also the diameter of nanotube. You actually can create almost unlimited number of code in this case. But previously it has just a silica nanotube. So for the separation and rinse those some other stuff after to look at the clear images of shape-differentiated nanotube in the microscope, and which is not really automated. So what we were trying to do was automation. So using this simply magnetic shape-differentiated nanotube. And we apply the magnet, and now what we're going to do is we put these whole systems in the microfluidic system, and now each nanotube can interact with analyte. Now we rinse them off, and then simply we can detect. That is our concept, and these are the magnetic nanotubes, shape-differentiated nanotube. This is one of the examples that we looked at, which is AFP sensing here. And these are all automated. And then this only BMN2 that has antibody that can interact with AFP only lights up in here in this case. So as a conclusion, you look at here, and I think I should stop here. And we'll be I think I'm gonna have to stop here.